Hello and welcome back. This is the second segment of a very large chunk of material titled Cells and Movement Across the Plasma Membrane. In this second segment, I will talk about the basic structure and function of the plasma membrane, and that is important in understanding transport mechanisms, which I'll begin here. And in the third segment, we'll pick up with transport mechanisms and continue from there. In this second segment, we will cover slides 44 to 103. And in the third segment, we'll finish transport mechanisms. So as I hope you recall, the plasma membrane is what we call the cell membrane that covers the exterior surface of all cells. And I don't just mean animal cells, and humans are animals. I don't just mean all eukaryotic cells. I mean all cells. Bacterial cells, archaea cells, plant cells, fungal cells, etc. So it's important to understand the structure of the plasma membrane, meaning what types of molecules it's made out of, so that you can then understand its function. In previous lectures, we have talked about atoms and molecules and the 4 classes of biological molecules and some of the properties of water. And understanding those molecules will help you understand how the plasma membrane behaves. And the plasma membrane is a key regulator of cellular function. In these images, we see plasma membrane. In the image on the left, that's a photograph taken using a transmission electron microscope. And we can see the tiny, tiny little plasma membrane there. And then, of course, on one side of the plasma membrane is intracellular fluid. Here it's labeled as cytosol. And then on the other side of the plasma membrane is extracellular fluid. So the plasma membrane is a barrier between intracellular and extracellular. And then in the artwork on the right, this is a representation of the basic structure of the plasma membrane, where a good starting place is to identify extracellular fluid. And in this representation, that's at the top in blue, and intracellular fluid, and it's sort of in that tannish color. And the plasma membrane separates the two. Molecularly, the plasma membrane is composed of a foundation of phospholipids, a double layer of phospholipids, also known as the phospholipid bilayer. And into that phospholipid bilayer are embedded cholesterol, proteins, and a few other types of molecules that we'll talk about. But definitely the foundational materials here are phospholipids, proteins, and cholesterol. Understanding the molecules that make up the plasma membrane enables you to understand the functions of the plasma membrane. And the plasma membrane has many functions. I have bullet pointed the 4 most important functions for our course, Human Physiology. So the first bullet point is that the plasma membrane maintains the chemical composition of the cytosol by selectively allowing materials to enter or leave the cell. I've mentioned this before as part of homeostasis. All cells, human cells, bacterial cells, everything in between, they maintain homeostasis, and one aspect of cellular homeostasis is maintaining a relatively constant composition of the cytosol. And the way cells do this is by allowing some materials to enter the cytosol and other materials to leave the cytosol. Bullet point number 2, plasma membranes have receptors that allow that cell to detect and respond to extracellular signaling molecules. And I touched upon an example of this when we were talking about transcription factors as part of protein synthesis in an earlier lecture. In that example, we saw that a signaling molecule bound to a receptor that was part of the cell's plasma membrane And that binding of the signaling molecule to the receptor initiated a cascade of events inside that cell that gave rise to a transcription factor, which regulates protein synthesis. So that came up in the context of protein synthesis, but it again comes up in the context of basic functions of the plasma membrane. Moving to the third bullet point, plasma membranes bear cell identity markers. So you're a collection of roughly 30 trillion cells. How come your cells don't get all mixed up? How do we keep kidney cells separate from heart muscle cells, separate from stomach lining cells? Each type of cell in your body bears a unique biochemical signature, and this signature is made up of molecules that are part of the exterior coating of the cell, therefore part of the plasma membrane. And then the fourth bullet point says, a function of the plasma membrane is to form structural junctions to neighboring cells and neighboring extracellular material. So there are few cells in our few cell types in our bodies that don't form structural tissues. Like red blood cells, for example, they're free floating. Red blood cells are not physically attached to any other type of cell. But the vast majority of cells in your body occur as part of a solid stationary tissue. And in order for these tissues to have structural integrity, their plasma membranes need to be connected to each other. And there are proteins in the plasma membrane that form these connections. So we'll talk more about the structure and function of the plasma membrane, taking care to hit on each of these bullet points. I've said it, how many times have I said it so far? Form follows function. So we need to understand the form of the plasma membrane so that we can more thoroughly understand the function of the plasma membrane. So let's start thinking about the form. And by form, I mean molecules. What molecules make up plasma membranes? So here are the most basic foundational materials of a plasma membrane. There is a double layer of phospholipids, and embedded in that double layer of phospholipids are proteins as well as cholesterol. So there's a phospholipid bilayer, there are proteins that can be associated with just the extracellular side of the membrane or maybe just the intracellular side of the membrane. There are also transmembrane proteins, that where trans means across. These transmembrane proteins go all the way through that phospholipid bilayer. And then there are scattered molecules of cholesterol, and cholesterol is a lipid, and that cholesterol is embedded inside that phospholipid bilayer. So phospholipids, I hope you remember, are amphipathic. And an amphipathic molecule is one that has both hydrophilic and hydrophobic regions. And so of this phospholipid bilayer, the exterior, both the extracellular surface and the intracellular surface, those pink heads that are shown here, each phospholipid, as is drawn here, has a pink head and two kind of greenish tails. Those pink heads are all hydrophilic. They include more polar covalent bonds, so they play the magnet game with water. And of course, these heads are surrounded by water molecules. Outside the cell, mostly water molecules. Inside the cell, mostly water molecules. And then we can look at those little green tails of each of those phospholipids. Those are made up of fatty acids, and they are hydrophobic. So when we talk about hydrophilic regions, hydrophobic regions, and amphipathic molecules, this is what we're talking about, and it matters a lot when it comes to the plasma membrane function. So we'll talk more about this. So that thick hydrophobic region in the middle of a phospholipid bilayer, that actually is a barrier. It's very difficult for a water-soluble substance to cross the plasma membrane. There's this fatty layer in the middle, and you remember, in general, fatty things are hydrophobic, and like dissolves like. So a substance that is hydrophilic, because it's either ionic or it has polar covalent bonds, is not easily soluble in something that's hydrophobic. So this relatively thick hydrophobic region in the middle does allow small fat-soluble substances to pass through quite easily. It's very inviting to a tiny hydrophobic substance like oxygen molecules, carbon dioxide molecules, steroid hormones. Steroids are another class of lipids. So all of those things are lipid soluble and easily pass through that phospholipid bilayer. But this hydrophobic region will act as a barrier to water-soluble things, charged things like ions. Don't readily pass through this barrier. In this artistic representation of the plasma membrane, there are additional layers of complexity. So let's start with what we know. In this representation, we see the phospholipid bilayer, we see proteins, and we see cholesterol molecules. So those three elements that we've talked about are present. Additionally, we see proteins that make up the cytoskeleton. So this must be inside the cell. These are cytoskeletal elements. We also see what I refer to as hybrid molecules. That's not the technical term, but it makes it easier for students to understand that there are lots of types of molecules that are made up of different aspects of the four classes of biological molecules. So for example, there's a glycoprotein here, and there are lots of different glycoproteins affiliated with the plasma membrane. And what a glycoprotein is a molecule that's part carbohydrate and part protein. So that's what I call a hybrid molecule. We can also use this drawing to look at the relationship between proteins and their location relative to the phospholipid bilayer. Some proteins are associated with just one side of the membrane, and they're called surface proteins. So down here, we have this protein here in blue, and it's only associated with the phospholipid heads on the intracellular surface, the inside surface of the plasma membrane. And then we have other proteins like this guy here, who's kind of stuck in but doesn't go all the way through the plasma membrane. And then we have, let's say, this guy over here. This is what's known as a transmembrane protein. And we'll probably spend the most time talking about a variety of transmembrane proteins. And what a transmembrane protein is it's a protein that goes all the way through the plasma membrane, all the way through the phospholipid bilayer. So a transmembrane protein contacts the extracellular fluid. It also contacts the intracellular fluid, and it passes through that thick hydrophobic region in the middle of the phospholipid bilayer. The reason why transmembrane proteins are so important for us is many receptors and ion channels, for example, are transmembrane proteins. So they'll come into play when we talk more about specific functions of the plasma membrane, pretty much all semester long. What we see here is one transmembrane protein embedded in the phospholipid bilayer, and this one protein is represented in purple. And let's start by reviewing what we already know about proteins. So I hope you remember that proteins are made out of long chains of amino acids, and I think it's the artist's intention to use these little purple balls, where each purple ball represents a single amino acid. And then when parts of the polypeptide chain twist and turn, maybe that's represented by these cylinders here. This part of the molecule is still made up of amino acids, but those amino acids are very folded, maybe into what's known as an alpha helix. And every protein has a terminal amino group and a terminal carboxyl group. And transmembrane proteins necessarily have to be amphipathic. Because they're amphipathic, they can be stabilized. They can be stable residents of the plasma membrane. If they were not amphipathic, they would easily pop out of the phospholipid bilayer. So I'll tell you that there are two different regions of this one molecule that are hydrophilic, that are water-loving, because their amino acids include more polar covalent bonds. Their amino acids more readily play the magnet game. So two regions of this protein tend to be polar. One region of this protein tends to be hydrophobic or nonpolar, and this is because there is an abundance of amino acids that have more nonpolar covalent bonds in this region of the molecule. So can you identify those regions? Which two regions are likely to be hydrophilic and which one region is likely to be hydrophobic? I hope you got this right. So the two hydrophilic regions are the part of the protein that extend into the extracellular fluid, and then the other part of the protein that extends into the intracellular fluid. Because extracellular fluid and the intracellular fluid, they're both mostly water. And then what's the one hydrophobic region of the molecule? It's the region of the molecule that we find embedded firmly in the phospholipid bilayer. So it's this region of the molecule. The amino acids here are going to have lots of non-polar covalent bonds. And so this allows this protein to remain stably embedded in this phospholipid bilayer. Plasma membrane proteins are super important for the function of the membrane. Here are three basic functions. Number one, plasma membrane proteins allow specific materials to enter or leave the cell, and we'll spend a great deal of time talking about ion channels, transporters, and pumps. Plasma membrane proteins can also act as receptors to which extracellular signaling molecules combine, and we will spend a whole lecture topic talking about cell signaling. And then, as I mentioned previously, plasma membrane proteins can also link neighboring cells to each other. So here are some examples. Here's a plasma membrane protein. It is a transmembrane protein. It is a transporter. More specifically, it is a glucose transporter. Glucose transporters, guess what they do? They transport glucose. It's a great name. Glucose transporters are a type of transmembrane protein that allow glucose to enter cells. Glucose is full of, mostly, polar covalent bonds. So glucose is largely hydrophilic. And so glucose has a hard time passing through the fatty acid middle of a membrane. And so in order for glucose to get into our cells, those glucose molecules need to pass through this transporter protein. Some membrane proteins act as receptors, and receptors have a particular three-dimensional shape that allow a very specific signaling molecule to bind. And so we can think about hormones and hormone receptors as an example. We'll use insulin as an example hormone. It brings us back to that example of diabetes as the body's inability to maintain homeostasis. And so I hope you remember that insulin is a hormone, a signaling molecule that's produced by the pancreas, and most of our body cells have receptors for insulin. So the receptor is in blue here, and the insulin molecule, the signaling molecule, is in red. And the receptor has a specialized three-dimensional shape into which only an insulin molecule will fit, so no other molecule can activate that receptor. And when the signaling molecule binds to its receptor, that activates a response on the part of the cell. A third function for plasma membrane proteins is to form physical connections between neighboring plasma membranes. So one cell can be firmly attached to its neighbor or to extracellular material. And so there are a variety of cell junctions that we'll talk about later. Right now, this is just an overview of, well, what do plasma membrane proteins do? And here, let's see, what color should we use? Let's use red. So here, we see a variety of plasma membrane proteins that anchor these two neighboring cells together. And then down here, we see a different set of membrane proteins that anchor this plasma membrane to what's known as a basement membrane. That's a structure that occurs between cells. So it's not really part of any one cell, it's extracellular. And we'll talk more about cell junctions down the road. Other plasma membrane proteins function as cell identity markers, and these are two examples of those quote hybrid molecules I referred to earlier. Again, hybrid molecule is definitely not a technical term. I just think it's helpful for students to understand that these molecules can be a mixture of different biological molecules. So glycoproteins are part sugar, part protein. Glycolipids are part sugar, part lipid. Maybe a better way to say that is they're part carbohydrate, part protein, part carbohydrate, part lipid. An example of a cell identity marker is a glycoprotein called MHC, where MHC stands for major histocompatibility complex. It's also known as HLA, human leukocyte antigen. So all of your cells bear MHC in their plasma membrane. And your MHC is unique. No two people have the same MHC unless you are genetically identical twins. And so MHC allows your immune system cells to distinguish between your cells that belong in your body and other cells that don't belong in your body, like bacterial cells. But they also present a challenge for tissue transplantation or organ transplantation. If you want to take the heart out of one person's body and put it into another person's body, the recipient's body may reject that donated heart because it has the wrong MHC. So this is just an example of a glycoprotein that functions as a cell identity marker. You have many different cell identity markers. And sometimes those cell identity markers are referred to as the glycocalyx. The glycocalyx refers to this carbohydrate-rich coating to the extracellular surface of the plasma membrane, made up of glycoproteins and glycolipids, where the glyco, the carbohydrate part, protrudes into the extracellular fluid. We can use this artistic representation to revisit or summarize some of the ideas that we've been talking about related to membrane proteins. So allow me to show you that some membrane proteins are surface proteins. They're associated with just one side or the other of the plasma membrane. Lots of membrane proteins are transmembrane proteins. They go all the way through. Some membrane proteins are glycolipids or glycoproteins, so they're part carbohydrate and part protein in this example. And we will talk a lot about membrane proteins, transmembrane proteins that increase the permeability of the plasma membrane by forming pores or channels that allow materials to pass through. So all the ways in which substances can cross the plasma membrane, those are called transport mechanisms. So if I'm walking down the hallway at school and Dr. Reyes says to me, "Hey Laura, what are you talking about in class today?" and I say transport mechanisms, he instantly knows that we're talking about the plasma membrane and all the ways in which different substances can possibly get across it. And so some materials require a membrane protein to get across and others don't. Some methods of crossing the membrane require the cell to spend ATP, so the cell might have to spend energy, and other methods do not require the cell to spend ATP or energy. This table comes directly from your textbook. Please don't memorize it. I'm using it as a tool to illustrate, to remind you that the composition of fluid outside the cell, extracellular fluid, is different from the composition of fluid inside the cell, intracellular fluid, or we can think in terms of cytosol. And both of them are watery solutions, right? Both of them are body fluids, But when we go solutes by solute, we can run down the list of solutes here and we can do the side-by-side comparison. Let's start with sodium. So if we look outside the cell, there's a much higher concentration of sodium ions relative to inside the cell. But the opposite is true for potassium. For potassium inside the cell, we have a much higher concentration relative to outside the cell. And we can keep going on and on. So what's responsible for maintaining this exact composition of intracellular fluid? It is the plasma membrane. That's one of its major functions. There are many different transport mechanisms, many different ways in which a particular particle, a particular substance can get across the plasma membrane. And the biggest division, the two biggest categories, is, well, is it a passive mechanism or an active mechanism? Passive transport mechanisms don't require the cell to use ATP. They'll just happen given the free energy in the system. And we can contrast passive transport with active transport. Active transport mechanisms require the cell to spend ATP, so it's costly for the cell to do this thing. We'll start with passive transport mechanisms, and then we'll move on to active transport mechanisms, I think, in the next segment. So these are three passive transport mechanisms, and again, I'll remind you, the energy that drives this movement is already present in the system, and the cell is not required to use any ATP to make these happen. That's why they're called passive. Like, what does it mean when somebody's a passive person? Oh, she's a very passive person. She doesn't really tend to take action, right? She's not going to spend much energy to get something done. And so these are the three passive transport mechanisms: simple diffusion, facilitated diffusion, and osmosis. In order to understand all transport mechanisms, really, we need to start with an understanding of concentration gradients or a review of concentration gradients. And so if we're looking at the plasma membrane as a barrier between two different solutions, intracellular and extracellular, we say a concentration gradient exists when for a given solute, a given substance, there's a higher concentration on one side of the membrane and a lower concentration of that substance on the other side of the membrane. Here's a very simple diagram to illustrate that, where that blue and gold line down the middle is representing the plasma membrane, and our substance is purple dots. There's definitely a clear concentration gradient here. On the left side of the membrane, we have a higher concentration of purple dots, and on the right side of the membrane, we have a lower concentration of purple dots. So imagine that these purple dots can physically cross this membrane. Imagine that it's very easy for a purple dot to just go right across from one side to the other. These purple dots will diffuse from the area of high concentration toward the area of low concentration. Diffusion will ultimately eliminate a concentration gradient. Look at these panels. We'll start with the panel on the left, where we see a clear concentration gradient and a membrane that is permeable to these orange molecules of dye. If diffusion proceeds, more dye molecules will move from left to right, then right to left. And the two solutions on the left side and the right side of the membrane, both of those solutions will come into what's known as equilibrium. When the concentration gradient has been destroyed, in this case, we say that those two solutions are in equilibrium and there are equal concentrations of orange dye molecules in the solution on the right side of the membrane and the solution on the left side of the membrane. By graphing the concentration of solutes in two different solutions, we can watch two solutions come into equilibrium with each other. So there are two components to this image. Up top, we have a visual representation of two different solutions in two different fluid compartments, compartment 1 and compartment 2. And we see what happens as solute moves between those compartments from time A to time B to time C. And then down below, we can graph the concentration of the fluids in each of those compartments over time. So let's start with the images up top. We have our two compartments and they're separated by a semi-permeable barrier that allows the purple dots to cross. At time A, there's a steep concentration gradient, and purple dots will move by diffusion from compartment 1 to compartment 2, such that when we get to time B, now there are some purple dots also in compartment 2, and we have bidirectional movement of purple dots, indicated by both of those arrows. The longer the arrow, the greater the rate of diffusion in that direction. So at time B, we still have a concentration gradient. There's a higher concentration of purple dots in compartment 1 and a lower concentration in purple 2 in compartment 2. And so notice that the arrow moving from compartment 1 to compartment 2 is longer than the arrow moving in the opposite direction. So there is a bidirectional movement of solute. It's just happening in a greater rate as purple dots move from compartment 1 to compartment 2, and they move more slowly in the opposite direction. But then we get to time C. At time C, we have achieved equilibrium, where there are equal concentrations of purple dots in compartment 1 and compartment 2. Purple dots are still moving, diffusion is still happening, but it's happening at equal rates in both directions. And we can see that both black arrows there are the same in length. And then we can take that information and portray it graphically. So let's move to the graph below. On the horizontal axis, we have time, and we see our 3 time points labeled there, time A, time B, and time C. And then on the vertical axis, we have glucose concentration. And the green line on the graph represents the concentration of glucose in compartment number 1 over time. And the purple line represents the concentration of glucose in compartment number 2 over time. So let's look at time A. At time A, all of those glucose molecules are in compartment 1, and none of the glucose molecules are in compartment 2. At time B, some diffusion has occurred, and so there are fewer glucose molecules in compartment 1, and there are more glucose molecules in compartment 2. And then by the time we get to time C, there are equal glucose concentrations in both compartments. Diffusion can happen with 2 or more substances in opposite directions, where these substances will act independently of each other. So we have 2 different concentrations shown here, the image on the left. We have 2 different substances with 2 different concentration gradients. There's the concentration gradient for the dark, are they purple? The dark purple dots. There is a different concentration gradient for the orange dots. And the purple dots are going to move by diffusion, and the orange dots are going to move by diffusion. And notice that they're both going to come into equilibrium. So we're talking about diffusion, and specifically simple diffusion. Simple diffusion means a substance crosses through the phospholipid bilayer where there's no involvement of a membrane protein. So only certain substances are able to pass through that phospholipid bilayer. That's called simple diffusion, and all of the substances that can do that have the following properties. They are uncharged, so not ions. They are uncharged molecules, and they are small molecules, and they are lipid-soluble molecules. There's a list of the most common substances that we'll encounter that cross the plasma membrane by simple diffusion. Oxygen, carbon dioxide, small fatty acids, not the big ones, they're too big, but only the little ones, the short ones, steroid hormones, and then ethanol. That's a small fat soluble-ish water molecule. So we're talking about diffusion. That's the movement of a substance downhill or along its concentration gradient from an area of high concentration to an area of low concentration. So everything shown here represents a type of diffusion. Simple diffusion occurs when a substance goes directly across the phospholipid bilayer. And then these are two other types of diffusion. So they're also diffusion, but they require a membrane protein in order to allow the substance through the plasma membrane. And so here we have simple diffusion, and it's contrasted with facilitated diffusion. Facilitated diffusion is diffusion. A substance is moving from an area of high concentration to an area of low concentration, but it requires a facilitator, specifically a membrane protein. So again, facilitated diffusion occurs when a substance crosses the plasma membrane by passing through a membrane protein. Again, it's diffusion, so we're always going from an area of high concentration to an area of low concentration. And this membrane protein acts as a channel, a pore, a door, a gate. So there are two different types of membrane proteins that are involved in facilitated diffusion. And we'll talk about both types of proteins almost all semester long. There are ion channels, and I've added the word gated here just to really make it clear that ion channels are gated. The gate is either open or closed, but sometimes we'll just use the word ion channel, but I hope you remember that ion channels are gated, and I'll show you what that looks like in a moment. And then there are transporters. Transporters are not gated, and transporters are sometimes called carriers. So what are these things, and how do we distinguish between the two? So gated ion channels are gated, where that gate can be open or closed. I'll show you an image of that in just a moment. And ion channels are for ions. Only ions pass through ion channels. And most ion channels are specific. So for example, a sodium ion channel only allows sodium through. It won't allow calcium ions to pass through. It won't allow magnesium ions to pass through. So ion channels are specific, as are transporters. So a fence has a gate. And so if you can think of an ion channel as having a gate that's open or closed, when the gate is open, it means that an ion can diffuse through it. And when the gate is closed, it means that it is impossible for an ion to pass through that ion channel. And so how is this achieved? How can this gate be opened or closed? This is an example of a protein that undergoes a conformational change, a shape change, as part of that protein doing its job. So we see a gated ion channel embedded in this plasma membrane. It's green, and this ion channel has two different shapes. In one conformation, in one shape, that ion channel is closed. The gate is closed. And in the other shape, the second conformation, that ion channel is open. The gate is open. And when the gate is open, ions are allowed to diffuse along their concentration gradient. We will talk a lot about ion channels, particularly when we talk about cell signaling and when we cover the nervous system. So ion channels differ according to, number one, the type of ion that they allow to pass. Is it a chloride ion channel? Is it a calcium ion channel? Maybe it's a potassium ion channel. So ion channels are specific, and then they also differ in what causes the gate to open or close. And so perhaps number one here is the hardest to understand. But the first type of gated ion channel is called a leakage channel. And every textbook I've ever read describes leakage channels as randomly fluctuating between the open and closed position. So if you can imagine a door that flip-flops back and forth, open, close, open, close, open, close. I'm suspect of that. Very few things in biology are random, but every textbook I've ever read said leakage channels randomly fluctuate. I just want to give you that little caveat there. Number two, there are ion channels that open and close depending on whether or not a ligand is stuck to the channel. So a ligand is a generic word for a substance that binds to something else. So certain ion channels will open when a substance sticks to them, when a ligand sticks to them. I'll show you examples in just a second. And then we have mechanically gated ion channels. This is how lots of your touch receptors work, for example. A mechanically gated ion channel will open in response to a physical force like pressure or vibration. And then voltage gated ion channels will change shape. They'll open or close when there's a change in membrane potential, when the membrane potential reaches a certain voltage. So let me use some images to more fully introduce these different types of gated ion channels. We will apply each of these down the road. Remember, this is foundational material, and these ion channels will become important, particularly during the nervous system, but that's not the only time. So leakage channels. Leakage channels randomly fluctuate between the open and closed position. The example in this picture is a potassium ion channel. When the potassium ion channel, this protein, is in one configuration, it's closed, and the potassium ions are stuck on one side of the plasma membrane or the other. Nobody's diffusing anywhere. And then when the protein changes shape, randomly, it opens up. And now those potassium ions are able to diffuse according to their concentration gradient. This is an example of a ligand-gated ion channel where a ligand might be a signaling molecule like acetylcholine. That's a neurotransmitter that's important in the way your muscle cells work. So acetylcholine will come up later as an important signaling molecule, specifically a neurotransmitter that opens up ion channels. So using the image on the left, there is no acetylcholine stuck to the ion channel. So the ion channel is in the closed position. The gate is closed and no ions can flow through it. But then when two molecules of acetylcholine, two molecules of ligand bind to the ion channel, now we're going to the image on the right, the ion channel opens up and these ions can diffuse according to their concentration gradients. This is an example, this is an unusual example of an ion channel, and it's true. There are some ion channels that do allow different types of ions to pass through. Most ion channels are specific for one type of ion. This is a rare example of an ion channel that does in fact allow sodium, calcium, and potassium ions to all pass through, but it is the exception, not the rule. Here's a representation of a mechanically gated ion channel. And let's see, lots of your touch receptors in your skin that allow you to sense pressure and light touch and vibration act this way. So this transmembrane protein, this ion channel, is closed until it's acted upon by a physical stimulus. That physical stimulus, that touch, that pressure, that vibration causes the protein to change shape. I know that's really cool, right? And when that protein changes shape, that gate is going to go from the closed to the opened position. And then we have voltage-gated ion channels. Voltage-gated ion channels change shape when the membrane potential changes. When that charge difference across the plasma membrane is altered, that protein will then change shape, and the shape change either opens or closes the gate. We will talk a lot about voltage-gated ion channels when we talk about how electrical signals are generated and transmitted in neurons as well as muscle cells. So ion channels are specific. A typical ion channel only allows one type of ion to pass through. It is far less common, but it does exist, that there are ion channels that allow 2 or 3 different types of ions to pass through, but they are far more rare. We are in the middle of talking about facilitated diffusion, And there are two general categories of proteins that allow substances to pass the plasma membrane by facilitated diffusion. We just finished talking about ion channels, and now we're ready to move on to the carriers, also known as transporters. And so transporter proteins, they don't allow ions to cross, but rather they move polar or hydrophilic molecules. And let's take a look at how they work. We'll go back to this example of a glucose transporter, and we'll talk about glucose transporters when we get to the kidney, and as well as in the digestive system is another place where glucose transporters might come up. So glucose is, I'll remind you, glucose is a 6-carbon molecule, and it is polar. So it plays the magnet game. It is hydrophilic. And as a hydrophilic substance, it cannot cross the plasma membrane by simple diffusion. It can't go through that phospholipid bilayer, but it needs a membrane protein to carry it across. This is why it might be called a glucose carrier or a glucose transporter. So how transporters work is they also undergo a conformational change, so a shape change that a protein undergoes as part of that protein doing its regular job. So this protein, the yellow protein that you can see in this image, it has two different shapes. And when the protein is in this shape, shown here, when the protein is in this shape, it's ready to collect a glucose molecule from the outside, from the extracellular fluid. A glucose molecule has the perfect shape to fit into that little nook in the glucose transporter. And then as soon as a glucose molecule fits into the nook, guess what happens? The protein changes shape, and that glucose molecule is released on the other side of the membrane. So this is how glucose gets into our cells. Our cells all have glucose transporters in their plasma membrane. Here's a question for you. Why can't ions cross the plasma membrane by simple diffusion? In other words, why can't an ion go directly through the phospholipid bilayer? What's the major reason? Ions are charged, and that phospholipid bilayer does not allow charged substances to pass through. So we need a gated ion channel to allow our ions to cross by facilitated diffusion. Our next mechanism of passive transport is called osmosis. And osmosis always refers to the movement of water. Nothing else moves by osmosis. If you hear the word osmosis, we're always talking about water molecules moving from point A to point B. And here's a catchy phrase for remembering how osmosis works. Where the solutes go, water flows. Water molecules will always move from the lower concentration solution to the higher concentration solution. So you can think about the water molecules chasing after the solute molecules or the solute particles. This is a common activity that's done in general biology classes so that students can really see osmosis occurring. So the activity begins when students take a beaker like this, where there are two different compartments in the beaker. There's the compartment on the left and the compartment on the right. And what separates the two compartments is this barrier. It's a selectively permeable barrier. And it's usually made out of dialysis tubing, this plastic that has tiny pores in it, where those pores are big enough to allow water molecules to pass through, but they're not large enough to allow sugar molecules to pass through. So it's a selectively permeable membrane, and the students add to the compartment on the left, they add a high concentration sugar solution. So if we have time, they make this sugar solution, they dissolve sugar in warm water, we let that water cool, and that's our high concentration sugar solution. And then in the compartment on the right, they add a low concentration sugar solution. So they add less sugar per volume of water. And we let that solution cool, and then we add each solution to each compartment. And at the start of the setup, notice that the level of fluid in each compartment, in the two compartments, is equal. There's an equal volume of fluid on the right side and the left side of this barrier. And then we have the students place the beakers at the back of the room, and we move on to something else. And then sometime later during the lab period, I have the students go check out their beakers and look at what happened. And they may be shocked and awed by the change that they see. Now there's clearly an unequal volume of fluid in each compartment, and this is caused by osmosis. We know the barrier is permeable to water, but it's not permeable to sugar, and water follows solutes. More water molecules moved out of the low concentration solution into the high concentration solution. You can think about nature abhors a gradient. This is nature's attempt to undo the concentration gradient. Glucose molecules can't pass through this membrane, right? It's semi-permeable. Water molecules can pass through, sugar molecules cannot pass through. And so how do we achieve equilibrium? How do we make these two solutions equal in concentration? Water is the only thing that can move. The water molecules will move from the area of low concentration to the area of high concentration. And that causes the volume of fluid in one compartment to go up and the volume of fluid in the other compartment to go down as more water molecules leave the compartment on the right and move over to the compartment on the left. So how do water molecules get across the plasma membrane? You might be thinking, well, but Laura, you've been telling us that there's this large hydrophobic region of the plasma membrane, and that's true. There's this big, fat, rich middle of the plasma membrane that is a barrier to hydrophilic substances. And well, what's more hydrophilic or water-loving than water molecules? You would be right in your thinking. And our current understanding is that water can cross the plasma membrane by two different means, two different mechanisms. And I'll talk about the more confusing one first. So shown here in this part of the diagram, we think that water molecules can cross that plasma membrane because they slip through momentary gaps in between neighboring phospholipids. So those phospholipids on the extracellular side of the membrane and the intracellular side of the membrane, those phospholipids are not fixed or static. They're not rigid. They're bumping past each other. They're sliding around, and occasionally a little gap between two neighboring phospholipid molecules opens up. And of course, there's water molecules everywhere outside the cell, inside the cell. There's just so many water molecules. And so as these momentary gaps arise, water molecules slip through the phospholipid bilayer. There's another mechanism for osmosis across the plasma membrane, and that's shown here, this big blue protein here. It's a transmembrane protein. It's known as an aquaporin. Aqua meaning water, pore meaning hole. This is a water channel, or sometimes it's called a water pore. So it's a transmembrane protein that the way it's built, it specifically allows water to cross the plasma membrane. And the thinking on the left side of this diagram, where water molecules pass through the phospholipid bilayer, this is an earlier hypothesis. We still think it's true because osmosis happens even in the absence of aquaporins. So we still believe this to be the case. And this idea, the fact that aquaporins exist, was a much later addition to our scientific understanding. And the person to propose this, hey, there might be water channels, this person was the subject of derision. His ideas were poo-pooed by the scientific community until he gathered mounting evidence. And now there is very obvious, clear evidence of the existence of aquaporins, or these water channels. And by the way, the scientist who initially proposed the existence of aquaporins and have his ideas poo-pooed by the scientific community, eventually he was awarded the Nobel Prize in Physiology or Medicine for his work on aquaporins. So moving on, We need to think about the relative concentration of cytosol, so the concentration of fluid inside the cell, compared to the concentration of fluid outside the cell. These two fluids need to have equal overall concentration because osmosis happens. And if they have unequal overall concentrations, the cell will either gain water, it will swell, it could possibly burst, or in the opposite concentration arrangement, the cell could lose water by osmosis. It will shrivel, it will shrink, the technical term is crenate, and the cell will lose its functionality. And what we want here, when homeostasis is being maintained, the solution inside the cell has an equal concentration to the solution outside the cell. And in that case, the cell shape remains stable. So we'll explore this further. The relative concentration of the fluid inside a cell to the fluid outside the cell is important for maintaining cell shape. And we can use these red blood cells suspended in a fluid in a beaker to illustrate this point. So these red blood cells are suspended in what's known as an isotonic solution. Iso means same, and the property of the fluids that we're talking about is tonicity. So what's their overall concentration? And the two fluids that are being compared here are the cytosol, so inside the red blood cells, and the extracellular fluid, so that blue fluid in the beaker. When we take red blood cells and we suspend them in an isotonic solution, that might be a 0.9% sodium chloride solution. When we take some blood out of your body and we add it to a test tube that has 0.9% saline, it's also known as physiologic saline, we say that those cells are suspended in an isotonic solution. The two fluids have equal osmolarity. There is no concentration gradient between the two fluids. So there might be some osmosis, but there's no directionality to the net movement of water molecules. Some water molecules are going to move into the red blood cells. Other water molecules are going to move out of the red blood cell, back and forth, back and forth. But because there's equal movement in both directions, those red blood cells are going to retain their normal shape. Next, we can see what happens when we take those same red blood cells, but we put them into what's known as a hypertonic solution, hypertonic solution. So the hypertonic solution, shown here in a darker shade of blue, has a higher concentration of solutes than the solution inside the red blood cells. So these red blood cells are suspended in you can think of it as a saltier solution, if you like. And the helpful tip for remembering osmosis is where the solutes go, water flows, water chases solutes. So think about which of these two fluids has the higher concentration of solutes. That's where the water molecules will move. So a hypertonic solution has a higher concentration of solutes relative to some other solution. So in this case, the other solution is the intracellular fluid. So the extracellular fluid has the higher concentration of solutes relative to the intracellular fluid. Water chases solutes. So there will be a net movement of water molecules from inside the cell to outside the cell. So these cells are losing water molecules by osmosis. The cells shrink. And when these cells shrink, they take on a spiky appearance, and that's evidence of the cytoskeleton. And then finally, we can add the opposite scenario. So if we take normal red blood cells and we suspend them in a hypotonic solution, so a very dilute watery solution, the cells take on water by osmosis. Where the solutes go, water flows. So when we compare our two solutions here, intracellular fluid versus extracellular fluid, we find the higher concentration of solutes inside the cells. And so water will move by osmosis toward the solution that has the higher concentration of solutes. And the cells will swell, as shown here in this image, and possibly burst. If the extracellular fluid is very hypotonic, very dilute, then the cells will take on so much water that they burst or they lyse. They split apart and they're no longer cells, they're no longer living things. Because osmosis will happen, the concentration of solutes in the cytosol and in the extracellular fluid need to be regulated. The solute concentrations in intracellular fluid and extracellular fluid, they're both physiologic variables, and sometimes we use the word controlled conditions that are governed by homeostasis. If homeostasis should fail, if one of those two solutions should become too concentrated, or the opposite, should have not enough solutes, then the cells, depending on the directionality here, the cells will either swell and possibly burst, or they'll lose water by osmosis and crenate, they'll shrivel up, and they won't be able to do their jobs adequately. So again, we've seen this table before. It comes right out of your textbook, and it allows us to compare the concentration of a variety of solutes in extracellular fluid with the same solutes in intracellular fluid. And as we saw previously, we just looked at sodium and potassium as examples, the differences can be quite stark. So there's a huge difference in the concentration of sodium outside the cell versus the concentration of sodium inside the cell. Again, a huge difference just in the opposite direction for potassium. There's far more potassium inside the cell than there is outside the cell. And we can go down the list, and there are big differences for most of these solutes. But the overall total concentration or osmolarity of each of these fluids turns out to be the same. So body fluids have many different solutes, and we can consider the concentration of one particular solute like sodium, but often we need to consider the concentration of all the total solutes, of all the particles that are dissolved in a particular body fluid. And that's when we need to talk about osmolarity. So osmolarity is a measure of the concentration of all solute particles in a solution. So let's think about these two examples. A 1 molar solution of glucose, where glucose is the only solute, a 1 molar solution of glucose has a concentration of 1 osmole. That's 1 osmole per liter. There's 1 mole of glucose in a liter of solution. You can think about it as 1 mole of particles, where each particle is a molecule of glucose, 1 mole of particles in a liter of water. That is a solution of 1 osmole. And then the second bullet point, we can think about a 1 molar solution of sodium chloride as having a concentration of 2 osmoles. It's 2 osmoles per liter. What happens when we take sodium chloride and we add it to water? Sodium chloride dissociates into sodium particles and chloride particles. So for every 1 mole of sodium chloride that we're adding to a liter of water, we're actually adding 2 moles of particles. There are sodium particles and there are chloride particles. So the osmolarity of a 1 molar solution of sodium chloride is 2 osmoles. Also, sometimes it's also said as 2 osmoles per liter. Molarity and osmolarity are two different ways of expressing concentration. And we can use this diagram to help us understand how to convert from molarity to osmolarity. So we'll go through these 3 different solutions, starting with the solution on the left. It's a 0.2 molar solution of magnesium chloride. So that means we take 0.2 of a mole of magnesium chloride and we add that to a liter of water. Magnesium chloride is an ionic compound. So when that ionic compound goes into solution, each original molecule of magnesium chloride will separate into 3 different ions. 1 magnesium ion, and 2 chloride ions. So for every 1 molecule of magnesium chloride, we get 3 particles in solution. And so we can look at the 2 different ways to express the concentration of this solution. It is a 0.2 molar magnesium chloride solution, but we can convert that to osmoles by multiplying by 3, because every magnesium chloride molecule, when it goes into solution, gives rise to 3 particles. So now we have a 0.6 osmole solution. Let's do another one. In the middle solution, our solute is sugar. I'll just insert the word glucose. Glucose is one type of sugar. And we have a 0.5 molar sugar, or a 0.5 molar glucose solution. That means we took half a mole of glucose and added it to a liter of water. And when glucose goes into solution, it doesn't give rise to different particles. Glucose is not ionic, rather it has polar covalent bonds, and those covalent bonds remain intact. So when a glucose molecule goes into solution, that whole molecule becomes surrounded by water molecules. So every glucose molecule that goes into solution gives rise to, well, just one glucose molecule. Nothing separates. And so the conversion between molarity and osmolarity is easy. There's nothing to multiply. A 0.5 molar glucose solution has an osmolarity of 0.5. The particle number doesn't change. Now let's think about our third solution on the left, where it has a concentration of magnesium chloride of 0.2 molar, and the concentration of glucose is 0.5 molar. How do we determine the osmolarity of this solution? In other words, what's the overall concentration of this solution when we don't distinguish among the different solutes? How do we get to 1.1? So we have to think about, for each solute, how many particles arise when that solute goes into solution. So as we previously talked about, magnesium chloride gives rise to 3 particles, and glucose, or sugar, just stays 1 particle. So we have to take our 0.2 molar magnesium chloride and multiply by 3, those 3 particles, to get 0.6. And we take our 0.5 and we just keep that at 0.5. You can think about it as multiplying by 1. That'll give you the same result. And so 0.6 plus 0.5 equals 1.1 osmoles. Here's a quiz. Can you answer this question? What's the osmolarity of 1 liter of solution that contains 1 mole of glucose and 1 mole of sodium chloride? I'll give you a moment to think about it. The osmolarity of this solution is 3. 1 mole of glucose will give rise to an osmolarity of 1 because that glucose remains intact. And the 1 mole of sodium chloride will contribute 2 osmoles to the solution because sodium chloride is an ionic compound and gives rise to 2 particles. So 1 particle produced by glucose and 2 particles produced by sodium chloride will give us an osmolarity of 3. So the osmolarity of both intracellular and extracellular fluid is roughly 300 milliosmoles, where a milliosmol is 1,000th of an osmole. In reality, it's closer to 280, but 300 is a nice round number, and it's used throughout your textbook, so we'll work with that. So the 2 solutions that we see here, intracellular fluid and extracellular fluid, they have different compositions, as we've seen previously in the table. But overall, they have equal osmolarity. And because the osmolarity is equal, this allows our cells to maintain their normal size and shape. So they don't gain too much water and they don't lose too much water by osmosis. There's a quiz question. In this state, is there any movement of water across the plasma membrane? Is there any movement of water? The answer is yes. Water molecules do continue to cross the plasma membrane, but they move in opposite directions at equal rates. These 2 solutions are in equilibrium. So osmosis is happening, but there's no net movement of water molecules. So here we have a review of some vocabulary that I introduced previously, and now I hope it's easy to apply 3 related vocabulary words at the bottom. So previously, I mentioned the terms isotonic, hypertonic, and hypotonic. And now we can think about the words isosmotic, hyperosmotic, and hypoosmotic. An isotonic solution, as we see here in panel A, causes cells to neither take on additional water nor lose water. So our cells maintain their original shape. We can also think about these cells as being suspended in an isosmotic solution. An isosmotic solution has an osmolarity of 300 milliosmoles. In the middle panel, panel B, we see these cells suspended in a hypertonic solution. A hypertonic solution causes cells to lose water by osmosis. And so the cells here are shriveled. The technical term is crenated. These cells, we can also talk about these cells in panel B as being suspended in a hyperosmotic solution. A hyperosmotic solution has an osmolarity greater than 300 milliosmoles, and so water will leave by osmosis. And then in the third panel, we see cells that are suspended in a hypotonic solution. And a hypotonic solution causes cells to gain water, and those cells will swell and possibly burst. We can also talk about the cells in panel C as being suspended in a hypoosmotic solution. A hypoosmotic solution has an osmolarity of below 300 milliosmoles, so it's more dilute than intracellular fluid, and the cells will gain water by osmosis. This image is from your textbook, and we can use that to reinforce our understanding of what happens to a normal cell when it's suspended in 3 different solutions. So we'll take the normal cell and notice that the concentration of the intracellular fluid, the concentration of the cytosol, is 300 milliosmoles, and notice the size of that cell up top. The size is representing normal cell volume. And we take that normal cell and then we'll plunk it into the solution that's on the left. We'll plunk it into a hyperosmotic solution. How do we know it's a hyperosmotic solution? Well, we can look at the osmolarity. And according to the label here, the osmolarity of this solution is 400 milliosmoles. So there's a higher concentration of solutes in this solution than there are in the solution that's inside the cell. And so this is also a hypertonic solution because it causes the cell to lose water and shrink. Next, we can take our normal cell volume, our normal cell, and put it into an isosmotic solution, also known as an isotonic solution. And we know that this is an isosmotic solution because our reference point is always 300 milliosmoles. And so the osmolarity of the solution inside the cell is equal to the osmolarity of the solution outside the cell. Osmosis is happening, but the cell neither gains extra water or loses additional water, and so the cell maintains its normal shape. So this is an isotonic solution. And then finally, we can take our normal cell and plop it into a hypoosmotic solution. How do we know it's hypoosmotic? Well, it's labeled here. The solution outside the cell has an osmolarity of 200, and 200 is lower than 300. So in a hypoosmotic solution, cells will gain water. That's what makes this also a hypotonic solution. And so we can see that the cell has increased in size as osmosis proceeds, more water enters the cell. It's a good time to take stock of where we are. We are talking about transport mechanisms. Transport mechanisms refers to all the ways in which a particular substance can enter and leave the cell. Our 2 big categories of transport mechanisms are passive mechanisms and active mechanisms. And we've just finished talking about the 3 types of passive transport: simple diffusion, which means right through the phospholipid bilayer; facilitated diffusion, which means through a transmembrane protein like an ion channel or a carrier, and then osmosis, the movement of water across the plasma membrane. And these passive transport mechanisms all rely on concentration gradients. Where we will go next is to discuss the mechanisms of active transport. And all these mechanisms of active transport, what they have in common is that they require the cell to spend ATP to cause the movement of a substance across the plasma membrane. And we will pick up there in segment 3 of the cells and movement across the plasma membrane lecture topic. So where we pick up in the next segment is to finish transport mechanisms. We'll pick up with active transport and go from there. Thanks for watching.